discuss with microsporogenesis. As you all know that anther is the male reproductive part of the flower, and here the microsporogenesis or the formation of male spores or the microspores will take place in the anther. Anther, you all are knowing that this is the male reproductive part, consists of mainly three parts. This is the anther, bilobe anther, then this is filament, and here connective. Now this anther is bilobe structure with filament and connective. When we will cut after the maturation, the third pole of the flower, that is anther or the male reproductive structure of the flower, will undergo process of microsporogenesis. When you will cut the transverse section of this part, that is bilobed anther, it will appear like this, four-lobe structure, because two lobes present in both the anthers. So in this way there are total 4 lobes are there. Now inside this lobes 4 sac-like structures are known as microsporangium. This sporangium, all these sporangium is a spore-like sac-like structure which will give rise to the microspore or the pollen of the plant. Now the tissue which is present inside this microsporangium, which are compactly arranged tissue, and they will activate, they will act as pollen mother cell or microspore mother cell. These pollen mother cell will enter into the process of microsporogenesis. Then we will study about this structure, enlarged structure of this transverse section of this microsporangium. We will get 4 layers here. 4 layers. Outermost layer is known as epidermis. Then second layer beneath the epidermis, endothelium is there. Then third layer, and lastly the 4th layer, that is tapetum. Now tapetum is the highly nutritive tissue, and these 3 upper layers are protected in nature. We'll see the diagram in detail. This PMC, that is pollen mother cell, or MMC, that is microspore mother cell, will enter into the process of microsporogenesis. For that, the cell will undergo the type of cell division known as meiosis. You all know that after the meiosis, at the end of the meiosis, 4 daughter cells are produced. Here also, it will give rise to tetrad, microspore tetrad, and all these 4 cells are haploid in nature. That means N or half number of chromosome is present. That's why it is known as haploid microspore. So from one PMC will get 4 haploid microspore which are present in the tetrad form. All of these 4 microspore will now give rise to the pollen. It is also known as pollen or male gametophyte. Now this pollen grain, all these pollen grains having the different structure externally and internally, that we'll discuss about the structure of the pollen grains. Then you will observe this pollen grain. You can see externally it is rough structure, generally circular in outline, having rough structure from outer side, and it is consists of 2 layers. Outer layer which is rough, irregular, and inner layer which is smooth. This outer layer is made up of sporopollenate, which is very hard, tough covering, and inner one is made up of cellulose and pectin. Now this outermost layer, which is so tough that it will not give entry of anything. That's why are present, from that germ pores only the pollen tube will come out. It is resistant to different types of chemicals, acids, etc. Now two germ pores are present generally in the monopod plants and 3 germ pores present in the dipod plants, which will give passage for the pollen tube during pollen germination. And inside this cell, the cytoplasm is there with prominent nucleus, and this nucleus will undergo, that means this cell will undergo division, that is mitosis critical, and then 2 cells will form. Bigger one is the vegetative cell and the smaller one is the generative cell. This vegetative cell is highly nutritive and this generative cell will give rise to the male gamete later on. This generative cell, again it will undergo one more mitosis to give rise to 2 generative cell, but the timing of this division of generative cell will be varying from plant to plant. In some plants, this at 2 cell stage, that means 1 vegetative and 1 generative cell, at this stage only the pollen will be pollinated or the stigma of the flower. But in some plants, before the pollination process only, the generative cell will divide into 2. That means at 3 cell stage, the pollen will be deposited on the stigma of the flower.
In this figure, you can see the transverse section of microsporangium. This having 4 lobes. These are the 4 lobes of microsporangium, and this is showing the enlarged view of this microsporangium. It is consisting of 4 layers. Outermost layer is known as epidermis. Then after endodermis, third layer is the middle layers, which is of 2-3 layered, and these 3 layers are protective in nature. And the innermost layer is tapetum, which is highly nutritive, and out of this, inside from here, this is the microspore mother cell. Some of the cells becomes activated as microspore mother cell or PMC, pollen mother cell, which will later on give rise to the microspore or the pollen or the male gametophyte of the flower. After formation of this male gametophyte, after meiosis and mitosis, you can see this is the diagram of a pollen grain. This is the external view and this is the internal view. Externally, it is consisting of 2 layers. Externally, outermost layer is known as exine and inner layer is known as intine. Exine is very hard covering, having 1, 2 or 3 pollen germ pores, and this is highly resistant to some chemicals and other things, made up of sporopollenin, whereas intine is made up of cellulose and pectin, which is thinner and it is not so hard. Now from inside you can see here that this in the cytoplasm, the cell is divided into 2, that is upper cell is known as vegetative cell and smaller one is known as generative cell. After this generative cell will undergo division, mitotic division, to give rise to 2 generative cell. In some plants at 2 celled stage it will be deposited on the stigma of the flower, and in some plants at 3 celled stage. Here you can see this pollen germination. When the pollen is deposited on the stigma of the flower, the pollen tube comes out through the germ pore.
Per pollen, 2 male gametes are produced and they are liberated or released inside the ovule during the fertilization process. Now, some of these pollens are also allergic for human as well as other organism also, and some of the respiratory disorders occurs in human due to these. For example, Parthenium grass, Gajar grass that we are knowing. Then these pollens are also preserved as a pollen bank, and later on they can be used by the plant breeders for the crop improvement program. That preservation is done in liquid nitrogen at minus 196 degree centigrade.